Morning, everyone. I think we've got the IT thing settled. Thanks for joining us for the Pioneers in Biomedical Research seminar today. We're excited to have Tiki Hayes joining us. She's an assistant professor of pharmacology at UCLA. Prior to this, she was a postdoc in the labs of Matthew Meyerson and Levi Garraway at the Broad Institute and Dana-Farber Cancer Institute up in Boston. Before that, she did her PhD at UNC Chapel Hill and Channing Durr's lab. She's published a number of highly impactful and innovative studies around genomic screening approaches to interrogate kinase structure function. She's an expert in receptor tyrosine kinases and KRAS signaling. Her work has been recognized by a number of grants and awards, including the NIH K99, the Damon Runyon Fellowship, the Burroughs Wellcome Fund grant, and the Eddie Mendez Scholar Award. I've known Tiki for over 10 years, and she's always been a real expert to me and someone that I refer to for experimental advice. So please join me in welcoming her. We're really excited to hear her talk today on variant functionality and mechanisms of resistance in cancer.
All right. Thank you for that. Oh, maybe a little higher. Thank you for that kind introduction, Kathleen. I want to thank everybody for the opportunity to share some of our science with the group. Okay, so my talk is entitled From Variant Functionality to Mechanisms of Resistance. And so the first thing I'm going to start with is my scientific journey. And so this is for the trainees. So if you have questions about this at lunch, I'm happy to discuss more. So I'm from Long Beach, California. And what I'm showing you here is a little Spartan because that was my first exposure to science, which is at my 8th grade science fair. From there, I went to UCSD for undergraduate studies, and I worked at the Burnham Institute for Cancer Research, or the Burnham Institute, not necessarily for cancer research, but I was interested in learning and having experience in the lab. And so I got an opportunity to work with a chemist who was designing small molecule inhibitors for phosphatases at the time. Then I took several gap years where I worked at the Ludwig Institute for Cancer Research, and I did yeast genetics. So I essentially went from looking at phosphatase inhibitors to yeast genetics. And then as Kathleen pointed out, I then transitioned to Chapel Hill, where I was really interested in understanding how proteins talk to each other, and how I assessed that was through understanding mechanisms of signal transduction. And then as a postdoc, I went to Boston because I was really interested in learning how to do functional genetics and genomics in order to answer these questions in a more systematic way. And then now I've come back to California, and now I'm back in LA, just up the street from actually where I'm from. Okay, so the talk today is going to entail 2 short stories. So one is going to look at variants of unknown significance in EGFR, and the other one is going to be a genome-wide resistance screen looking at MEK1/2 CDK4/6 resistance in NRAS mutant melanoma. So our lab is super interested in understanding the rules and mechanisms that govern signal transduction plasticity. And so what we really want to understand is how normal cells acquire mutations, and how those mutations lead to changes in downstream signaling cascades that regulate a number of important processes in cancer development of cancer. And then we really want to understand how we acquire mutations and how those mutations then affect downstream signaling cascades that lead to these changes in cancerous phenotypes. We're then also interested in understanding how then cells respond to targeted therapy, whether they're initially sensitive to targeted therapies or whether they're resistant, sort of de novo originally, or if they initially are sensitive, and how they acquire target resistance. And so how we study these is we're really interested in characterizing variants of unknown significance. So we really want to understand how mutations in these protein signaling cascades then give rise to disease cells. And so in terms of variant functionality, I like to break them up into 2 classes. So we have the first class, which are variants of known significance. So we know that these mutations are seen in patients, and we know that they're functional, and some of them, in fact, are sensitive to available targeted therapies. However, there's a number of mutations that continue to be uncovered with continuous human tumor sequencing, where we don't understand their functionality, and so we don't know if they could potentially drive disease, if they could, be a dependency, or if they're sensitive to available targeted therapies, or if we need to develop other targeted therapies. And so I'm going to tell you a story about EGFR, or the epidermal growth factor receptor, as a proof of concept for trying to systematically understand variants of unknown significance. So EGFR is receptor tyrosine kinase. It's a single pass transmembrane receptor tyrosine kinase that sits at the plasma membrane in a closed conformation. Once EGF ligand, as well as there are other ligands that can activate EGFR, allow for an open conformation. This open conformation allows for dimerization. Dimerization then leads to activation of a number of signaling cascades, and I'm showing 3 here. And so if we look at EGFR itself, it can be broken up into 3 domains. So we have the extracellular domain here, which can be broken up into 4 subdomains: domain 1, domain 2, domain 3, and domain 4. Domain 1 and domain 3, as you can see here, are important for ligand binding, whereas domain 2 and domain 4 are important for its dimerization. Then we have the single pass transmembrane domain. We have the juxtamembrane domain and the kinase domain, which gives the kinase its activity. And then we have the C-terminal domain, which is where we get a bunch of phosphorylation sites that then lead to docking of a number of proteins that then lead to activation of these downstream signaling cascades. And so why do we care about EGFR? Because it's frequently mutated in cancer. So these are data from cBioportal. Along the x-axis, we have EGFR positional information. Along the y-axis, we have number of mutations observed in cancer. I've thresholded it at 20, so we could see some of the rare mutations. However, some of these black dots are closer to thousands as such in TCGA. And so what you can appreciate, there are many black dots here. And so those black dots represent variants that we know their functionality. However, all these green dots, we see these mutations in patients, but we really don't understand how they function. And so EGFR itself can have a number of different types of alterations. So these include small and frame insertions and deletions, point mutations, amplifications, truncations, fusions, and splice variants. And so because EGFR is frequently mutated in patients, this has led to the generation of several small molecule inhibitors, which you can see here to the right, I guess to your left. EGFR also has a number of generations of antibodies that have also most recently been improved in combination with some of the tyrosine kinase inhibitors. And so when we think about the EGFR variant field, there are a couple of opportunities for improvement on what's already been established. So the current field for EGFR, most studies focus on patient-observed mutations, so they haven't been able to look at EGFR on our EGFR variant functionality on a systematic scale. And most of the studies have been using BAF3s, which are a marine cell line model that has taught us a lot about EGFR sort of sensitivity to targeted therapies, but hasn't taught us as much about EGFR biology. And so the opportunities in this space is to think about, a systematic variant approach for characterization. So doing this at scale and using other models outside of BAF3s to have a more physiological relevant system. And so the major question for us is, how can we systematically assess functional variants in proteins that are known to drive disease? So EGFR is a proof of concept, but you can imagine doing this in essentially any of your favorite targets. And so how you would think about that, so we have EGFR or your favorite protein. What you'll want to do is develop a library that has all of your potential mutations. And so you can do this via ORF or via CRISPR. I'm going to talk about ORF today, but there are CRISPR libraries that allow you to make endogenous mutations in the protein in your model system. The downside of that is you unfortunately need to have a PAM site. And if you don't have a PAM site near your mutation, you can in fact include it in the library. So it's sort of a pro and a con of how you want to think about or which kind of library you want to use. Then you want to come up with a functional assay that allows you to enrich for variants that promote growth, ideally, because it's harder, it's a lot easier to analyze upstream than it is a downstream. And so from there, you'll want to take your library, infect your cells such that you get one mutation per cell, look at representation of your population, and then treat with or enrich, however you want to enrich for your invariants, you can go ahead and do that. Collect them at the end of the enrichment process, compare them to what you started with, and then do some amount of computational analysis to determine which variants were enriched. And then you can have a bunch of applications for this, which can be basic if you really want to understand the functionality of your protein versus translational applications of do these variants cause resistance? And so our goal is to use these high throughput genetic screens to identify and map functional protein variants at scale. And so how does this actually work? So for the ORF overexpression system, we work with Twist Biosciences, which is a company up in San Francisco. And essentially they have proprietary site-directed mutagenesis that allows you to introduce a mutation or all mutations at any given amino acid position. So essentially you'll have 20 substitutions for every amino acid in your protein. So EGFR is a pretty big protein. And so our library ended up being fairly large with over 24,000 variants. And so how this actually works is you, in addition to the mutations that, so you additionally introduce silent mutations into your library. So when you overexpress it, you can then tell the difference between the endogenous EGFR and what you've in fact overexpressed. And then we have the high throughput site-directed mutagenesis. They give you linearized plasmid. And then from there, you clone your linearized plasmid into whatever you're screening platform. And so then you want to think about a functional assay that will allow you to enrich for your variants. And so one of the assays we use is PC9, which is an oncogene addiction model. So EGFR has an exon 19 deletion in the cell line, which is known to be an oncogenic driver, and that causes a dependency for the cell line. So if you treat with erlotinib or any other generation of EGFR TKI, you'll see sensitivity to the inhibitor. And so when we express our library, we essentially expect two different outcomes. We expect to find variants that are sensitive to erlotinib and variants that are insensitive to erlotinib. And so this assay itself is super imperfect because L858R, which is a known driver for EGFR, does not score in this particular screen. So this is a caveat of the screen. You replacement. And so our positive control is EGFR T790M, which is the known resistance mutation for erlotinib, so that should score on the screen. So essentially, as I mentioned previously, we'll take our library, infect cells, select out cells that incorporated our variants, set aside a plate for representation, then we treated with erlotinib for 10 days, and then collected those DNAs, and then we did the comparison to figure out which variants, in fact, were enriched. And so these are what the data look like. And so along the x-axis is EGFR positional information. Along the y-axis here, this is the average z-score enrichment. And so what you can appreciate is there are several peaks of mutations that are enriched. And so these two peaks here happen to correspond to the dimerization domain regions. And these are the actual amino acid substitutions. And so along the y-axis, these are the different amino acids themselves. Along the x-axis, we still have positional information. And what you can appreciate, there's a couple of bright red lines, spots here on the graph. So here, as well as here. And those two correspond to these peaks here. And those are actually cysteine mutations. And the other thing you'll also appreciate is there are a couple vertical lines here. And so those vertical lines are represented in the average z-score here, where you can see that there are several sites that no matter what the substitution is, you in fact get some amount of functionality. And so the next thing we wanted to know was how well was our library covered? So the number of variants we missed in the assay was about 1,600. And so we had about 93% of the library covered. So essentially, this graph, there's EGFR positional information on the x-axis, and these are the different amino acids on the y-axis. And the darker purple you are, the more variants you in fact missed. And so you can see that there are, sorry, the other way around. The darker you are, the more variants scored in that space. The lighter you are, the fewer variants that scored. So there are a couple sites where none of the substitutions scored in the screen, and so that could be a technical problem, or that could actually, in fact, be a biological question. And so from there, we wanted to align our enrichment data with data from somatic patient databases, such as Genie, and look to see if our variants, in fact, were seen in patients. And so these are data from Genie, and so the top graph here is GBM. The graph below it is lung cancer, and then the graph below that is all the above. So one of the interesting things is for EGFR in brain cancer, you can see that most of the mutations are in the extracellular domain. However, in lung cancer, most of the mutations are in the kinase domain. And so there's some interesting questions there about cell lineage and how mutations arise in different cell lineages. And so from there, we took a panel of uncharacterized variants from our screen, and we wanted to go ahead and validate them. So you can see the variants here on the right-hand side of the graph. And then what we did was in the first set of dose response curves, we went ahead and overexpressed a panel of these extracellular domain mutations that's scored in our screen and looked to see if they were able to drive resistance to erlotinib. And so as you can see, the red line here is C790M, our positive control. These here are our negative control, so our MOC, our LAC-C, and our wild type, and you can see a shift in sensitivity in the PC9s, a smaller shift in the HCC 4006, and then another shift in the HCC 827s. And we also did this for the intracellular domain mutations, where you can start to see a shift in sensitivity based on the dose response curves, suggesting that these variants, in fact, are able to promote growth in the presence of erlotinib, suggesting that they have some amount of functionality. And so the next thing we wanted to know is, well, what are the molecular mechanisms that are providing the ability of these cells to grow in the presence of drug? And so we wanted to look at downstream signaling cascades, is look at if there was any rebound of signaling downstream of our inhibitor block. And so the top graph here is the EGFR extracellular domain mutations in the PC9s. And so I'll point your attention first to our total EGFR, showing that we were able to overexpress EGFR, which is once again a caveat of the screen, but they were overexpressed to similar levels, so that makes our data more comparable across from variant to variant. And so what you can appreciate is if you look at phospho-ERK signaling, we can see a rebound in the amount of phospho-ERK, which was consistent with our growth phenotypes and both the PC9s as well as the HCC 4006. And so we wanted to understand if rebound phospho-ERK was also important for the intracellular domain variants, and we see the same thing, where we were able to see a rebound in the levels of phosphorylated ERK in both the PC9s and the HCC 4006. And so from these data, we wanted to understand if this was a dependency. And so what we wanted to do was then treat with an inhibitor that would inhibit MAP kinase signaling. So we use trametinib, which is a MEK inhibitor, and we assessed whether these mutations were able to grow in the presence of the combination. And so if you focus on the far panel here with the combo, you can see that there is a significant reduction in the amount of cell growth in the presence of the combination for both the extracellular domain mutations as well as the intracellular domain mutations. However, you can see a bit of residual growth, and so we wanted to understand if we inhibited phospho-ERK with trametinib enough to prevent growth. And so what we did is we did another Western blot to look at the levels of phosphorylated ERK. And so what you can see is, especially for the extracellular domain mutations, which you saw increased growth still in the presence of the combination, we still have some residual ERK, suggesting that there is maybe still some signaling to activate ERK downstream of both inhibitor blocks, which asks a question about the linearity of the RAS-MAP kinase signaling cascade. And so this is true for the extracellular domain variants as well as some of the intracellular domain variants. And so the next thing we wanted to do was understand if these variants were in fact sensitive to other generations of EGFR-targeted therapies. And so this is just a schematic of the development of EGFR small molecule inhibitors. And so essentially now we are currently, patients currently receive osimertinib, which is a third generation tyrosine kinase inhibitor. And what we've learned from these clinical data is that depending on what kind of mutation you have will dictate what kind of treatment you get. So if you have an atypical mutation, you're more sensitive to second generation inhibitors as opposed to the third generation inhibitor. And so we really wanted to understand our extracellular domain variants, which have a wild type kinase domain, so they're not sensitive to osimertinib because that drug specifically hits EGFR mutant kinase domains. Sorry, we have not looked into that yet. but that is something that we could definitely look into. So the next thing we did was we took some of the second generation inhibitors, specifically docomitinib, to understand whether we had inhibition. So what we've done is we once again did cell titer glow assay, where we have increasing amounts of docomitinib along the x-axis and cell viability along the y-axis. And T790M is our positive control because that causes resistance for this mutation as well. And so you can see from the colony formation as well as dose response curves, you do see sensitivity to docomitinib, especially at some of these higher doses. And that is consistent with loss of phosphorylated ERK as well as phosphorylated EGFR. And so the other thing we wanted to assess is there are variants of known significance in the extracellular domain that are seen in patients that we know, in fact, are functional. And so we wanted to understand whether these mutations were also sensitive to afatinib. So we took a panel of those mutations. And so what you can see similarly in this dose response curve, you can see sensitivity compared to the positive control, which is the T790M. And this is also consistent with loss of phospho ERK, and we also see this in the colony formation assays as well, suggesting that these mutations are in fact sensitive to afatinib. And so because most of the extracellular domain mutations, as I pointed out, were seen in GBM and not lung, there aren't actually any patient samples because patients haven't been treated with this sort of combination. However, there were studies using docomitinib and GBM that weren't particularly successful, but there is some interest in the fact in this particular mutation. So this G598V mutation, which we have 2 patients essentially who respond by essentially preventing the expansion of the disease, which was a lot for GBM since there aren't that many available targeted therapies. We had a second patient who started with a G598V mutation, but then upon post-treatment sequencing, found that mutation to be gone, suggesting maybe some sort of dependency for this particular mutation with these second generation inhibitors. And one of the really interesting things that we want to start focusing on is these rare extracellular domain mutations seen in lungs. So they are seen, and the question is, are they sensitive to maybe EGFR antibodies, or would they be more sensitive to second generation EGFR targeted therapies? So in this first part, I hope I've conveyed that we were able to identify and validate previously uncharacterized EGFR variants. Variants were able to overcome erlotinib treatment by upregulating MAP kinase signaling, and these mutations were partly dependent on MAP kinase signaling for growth, and some of these EGFR extracellular domain mutations were unexpectedly sensitive to docomitinib, which is a second generation EGFR inhibitor. Okay, so now I'm going to transition to the second part of the talk. We're going to look at genome-wide mechanisms of resistance to MEK and CDK4/6 in NRAS mutant melanoma. So if we go back to our overall schematic, we really are now talking about sort of this transition of cells that are sensitive, but what actually drives resistance in this context. And so when we think about resistance, I'm showing you 3 examples here, but there are far more examples of resistance, but you can think of sort of de novo resistance as the tumor just doesn't respond. You can think of on-target acquired resistance, and so generally speaking, that's a drug pocket mutation where you have a mutation in your protein of interest, and then you sort of have a clonal outgrowth in that capacity. Then there's off-target acquired resistance, which makes up a lot of resistance mechanisms that we still really don't understand, where you have this target sort of bypass, and so you can have multiple clones that are able to grow out in a similar tumor. And you can imagine that in some tumors, you might have acquired resistance and off-target resistance. So it's always more complex than we initially thought. And so RAS itself is made up of 3 isoforms, HRAS, NRAS, and KRAS. And as you can see here, they are all mutated in different cancer types. And so KRAS is a little bit different than NRAS and HRAS. That's based on its lysine stretch, which leads it to bind or interact differentially with the plasma membrane. And for KRAS, there are now available small molecule inhibitors that have reached the clinic. However, that's not as true for HRAS and NRAS to date. So essentially, we really wanted to understand NRAS mutant melanoma. And so what you can see here is approximately 30% of people who get melanomas are going to present with an NRAS mutation. And so how can you inhibit RAS? Generally speaking, as I talked about, there's now some inhibitors for direct inhibition. However, for the other two family members, you're really thinking about inhibiting membrane associations, synthetic interactors, post-translational modifications, potentially metabolism. However, downstream effectors have been the most well characterized and most effective to date. And so given that, there was a study performed about 10 years ago by Larry Kwong and Linda Chen's lab, where they went ahead and wanted to understand which pathway components could equal ablation of NRAS. And so essentially what they did was they took NRAS, they genetically ablated it, and then they treated with or in the mouse model, I should say, the apoptosis was driven by MEK and cell cycle arrest was driven by RB. And so given that, they came up with a combination strategy for NRAS mutant melanoma, which is treatment with trametinib and palbociclib. And so the goal of this project was to use high throughput genetic screens to clinically predict relevant mechanisms of resistance to this therapeutic strategy. Okay, and so when you think about resistance, you can think about your favorite target, not necessarily mine. And when you think about this, you can have sort of target dependent, as we talked about, so mutations in that protein that can cause resistance, or you can have target independent, so sort of like the genome wide, so whether you're thinking of the human Orpheome or CRISPR knockout screen, so looking for things that are able to bypass the inhibitor block. Similarly, you need a functional assay that your inhibitor essentially will inhibit variants that are enriched for proteins that are able to grow in the presence of your inhibitor. So for us, the first thing we wanted to do was identify NRAS mutant melanoma cell lines. And so what we did is we used data from Broad and Novartis and Marcotte. So what we can see here on the y-axis and the x-axis, the more negative you are, the more dependent you become. And so from there, we went ahead and took siRNAs against NRAS, and we went ahead and looked at cell viability over time, and then we also looked at colony formation, and were able to show that these cells were, in fact, dependent on RAS signaling. And so from there, we went ahead and set up a screen representation, treated for 21 days under these different conditions, and then looked for things that overlapped. And so these are data from the ORF overexpression results. And so what you can see is there are a number of different protein families that scored in this screen. And so things that were positive have a z-score greater than 2, things that were negative have a z-score less than 2. And so if we wanted to, if you are interested in something that's clinically relevant, which we were, we went ahead and went to see Bioportal to look at which of these proteins might actually be mutated in NRAS mutant melanoma, or at least altered. And so you can see here that there are a number of these signaling proteins that scored in our screen that in fact are seen in NRAS mutant melanoma. And then on your right-hand side, you can see the different types of mutations that are associated with those different proteins. And so from there, we want to go ahead and validate our findings. And so what we did was we overexpressed several of these mutations, so AKT1 to PI3 kinase mutant mutations that are seen in patients, as well as EGFR L5R, which is seen in lung cancer. And so what you can appreciate, so along the x-axis, we have trametinib increasing doses. Along the y-axis, we have increasing doses of palbociclib. And what you can see is compared to the GFP, you start to see this diagonal cell growth, suggesting that they are in fact resistant to the combination. And then this is also seen in the colony formation data as well. And so the next thing we wanted to do was understand if there was signaling that was driving this sort of resistance. And so what we've done is we took these cells and these cell lines expressing these different protein variants, and we treated them either with vehicle, trametinib alone, palbociclib alone, or the combination. And so essentially what we ended up finding was, if we focus here on S6 kinase, you can see a rebound in the amount of S6 kinase under all of these conditions, suggesting that we were able to overcome sensitivity by treating with this, able to overcome sensitivity by treating with this combination. And so it turns out that the PI3 kinase E545K mutation turned out to be clinically relevant. And so these are data from 2 papers that came out around the same time. So essentially there is a case study where they looked at this E545K mutation, and pre-treatment, you can see there wasn't as much staining, but you can see after they treated, you can see an increased amount of staining, suggesting that this mutation, in fact, did show up and led to the continued growth of the tumor. And then we also had a mouse model where they found the same thing, where essentially they treated with the combination. And if you focus here on S6 kinase in the resistance tumor, you can see a rebound of those, a rebound of S6 kinase. And so what this shows is essentially we can use these cell line models to predict mutations that might arise in patients clinically. Okay. And so this is the second part, summary from the second part. So we were able to identify and validate genome-wide resistance mechanisms to MEK and CDK4/6 alone in combination. It seems that the EGFR PI3 kinase AKT signaling cascade was able to drive resistance alone in a combination in multiple models of NRAS mutant melanoma. And this resistance resulted in a rebound of phosphorylated S6 kinase levels. And the PI3 kinase E545K mutation is in fact clinically relevant mechanism of resistance to this combination. And so the last thing I'll leave you with is, if you're a trainee and you're interested in a postdoc, or you're an undergraduate who wants to apply to graduate school, why come to LA? Well, let me tell you. So LA does have some greenery, not as much greenery as here, but it is nice. We have the beach, so this is Santa Monica, and this is Manhattan Beach. We have famous people and fun things to do, so if you like Insecure, this is actually where the apartment complex it was filmed. And so this is UCLA itself, so we have really strong basketball programs this year, so if you're interested in sports. We also have professional sports, the Lakers, Clippers, other football teams, the Rams, and the Chargers. And then we also, once again, even though we're a desert, we still have, nice, pretty ocean waters and things. Okay, so why not LA County? This is something that you guys could all relate to. There's a preponderance of traffic, so I'm sure at 5 o'clock today, you guys will experience that as well. And the other thing is something you experience here as well is the cost of living is pretty expensive. And so with that, I'd like to thank everybody who joined our small lab. So we've been around for about a year. And so if any of these things interest you, we're open to collaborations, as well as other people who want to join our group. And at this point, I'm happy to take any questions.
Hi, that was really nice way to test variants and everything. And so I'm interested more in birth defects, and I know EGFR is involved in, but it's in birth defects, but it's more loss of function mutations and or variants. And do you have a way in your assays, can you see the loss of function variants also? And have you correlated that with those results with syndromes that are caused by EGFR?
So the question was, do we see essentially dropouts, so things that essentially are dominant negatives that inhibit growth? And so yes, so it's not something I didn't actually, okay, this is probably the slowest way to do this, but we'll get there. Okay, yes, yeah, so we'll go here. All right, so yeah, so as was discussed, there are a bunch of things that in fact are dominant negatives, and so we actually found a site So there's a truncation of EGFR that's known to be a dominant negative, and so that specific amino acid actually was one of the missing variants in the screen. However, we did find a variant that was 2 amino acids smaller that in fact was a dominant negative. And so you can in fact find dominant negatives from these types of screens. And yeah, so yeah, so that could be a whole different class of things to look for. So EGFR itself also has a bunch of germline mutations. So I was talking about that earlier today. And these mutations, in fact, aren't disease driving, but they tend to cause resistance to some of the available targeted therapies. And so like that of itself is interesting. But to your point, something we could consider, which I briefly mentioned, is redoing this screen in a cell line that's maybe looking at more like transformation phenotypes, so you can look at things that more clearly fall out, and then maybe use computational tools to then align those with things that are actually seen in patients. Okay, so yeah. So the question was, have we looked at some of the truncation mutations? And so I have a project at an undergrad is actually going to start. So the nice thing about the screen is we include a bunch of stop mutations. And so we actually did get a bunch of truncations. And so the question is, we're going to then take these data and we're going to go into Genie or we're going to go into CBioportal and then see if any of those truncations are seen mutated in patients, and then maybe follow up on some of those things. So yeah, we're super interested in that. Why? All right, so the question is, based on the slide that's presented, why do we have these sort of different enrichment of variants in these specific domains? And so one thing I would point to is the fact that this assay itself is super imperfect. And so from one of the downsides of the assay is there are functional variants in this screen that are sensitive to erlotinib. So if we did sort of a different assay, what we might find is a slightly different landscape of enrichment. However, to get more to your point, what this might suggest is that many of the mutations that are needed for oncogene replacement that are insensitive to erlotinib do end up in the dimerization domains, which makes sense. So if you enhance dimerization, you likely will promote downstream signaling and growth. And in the kinase domain, we would actually expect most of the mutations, well, not most, or we would expect a subset of mutations to actually drive kinase sort of functionality. So you might see increased growth. However, many of them are sensitive to erlotinib. And especially in the C-terminal domain, I thought potentially there's a couple of phosphorylation sites that I thought might score in the screen that didn't. So there's like the 1068 that I thought might drive resistance, because then that could lead to binding of the scaffold proteins, which can lead to the downstream signaling cascades. Yeah, so the question was, how do we choose our dose treatments? So essentially what we've done is we did a bunch of dose response curves looking at the IC50 and GI50, and then looked at downstream signaling with these different concentrations. And then from there, we picked concentrations that weren't too toxic to the cell line, but also led to like pathway inhibition. She just spoke, just. Oh, we got a microphone.
I have two questions, like curiosity. One thing is your reference pool is time 0 of treatment or time end of treatment?
Yeah, so so how it works is you essentially infect a boatload of cells. So we're talking about like 1 billion cells you infect, and you expect to get maybe a couple 100,000 out. And then from there, you you count them and you plate them in T20 or T75s, and then you start dosing. And so that's the library point, is when you are plating to then drug.
Okay, so this is T0 of the treatment point you
Yeah.
are referring to. So now my question is, so if some of this mutation has hyperproliferation irrespective of the treatment, that you see is a positive hit here. So have you compared the pool, which is equivalent time to your treatment end time?
Yeah, so we actually did that. So we have a DMSO arm as well as an early time point arm, and we compared those across, and there weren't too many variants that grew significantly more in the DMSO control compared to the early time point. But it is something that sorry, can you repeat that?
All the cells you used, they still have the endogenous EGFR?
Yeah, so this is once again sort of a caveat of the system. So the question being about the endogenous EGFR in the system. So essentially the inhibitor should inhibit all the copies, but there's like 8 or 9 copies of EGFR in PC9. And so the idea being, if you treat the cells with an EGFR inhibitor, most of them will die. And so essentially what you're selecting for are variants that are insensitive to erlotinib and that can replace that endogenous exon 19 deletion signaling. And we don't treat long enough for those cells to develop resistance.
Yeah.
Okay.
Thank you.
Questions?
Mutational signature in lung, or like what, we talk about, why is APC mutated only in colorectal, but it's not like you get distinct mutations in APC and other tumor types. So what do you think is special about EGFR? That, I guess, the ligand, but like availability, what's driving that?
Right, so this question essentially is related to this slide. So where you see in GBM, we have a bunch of extracellular domain mutations, and in lung cancer, we have these kinase domain mutations. So the short answer is, it is unclear. What I can say is that it seems from clinical data, at least for EGFR lung cancer, we know that if you have an EGFR mutant lung cancer kinase domain mutation you treat with an inhibitor, they in fact are dependent. However, that is not as true, at least in cell culture models, for the extracellular domains in GBM. So the question then is more sort of a in GBM compared to lung cancer. So are there different ability? So essentially, in GBM, it doesn't seem to be as strong of a dependency. And so why is that compared to lung cancer, where you see this strong dependency? And so that's sort of one of the questions we want to get at. So we want to take some of these extracellular domain mutations that we see in lung, and we want to understand if they're in fact drivers. Because if they aren't drivers, that might just say, well, the extracellular domain mutations are important for initiation, but not maintenance. And so, yeah, so we are going to do those questions. And then we're also going to take some of the kinase domain mutations and hopefully put them in GBM cells, which there are. So there are some kinase domain mutations which are known to be functional in GBM, but they're rare compared to extracellular domain mutations.
So thank you so much, Tiki. Are there any more questions or any questions online?
I've got one more, beautiful science, but really depressing from a targeted therapy kind of perspective, potentially, so, just in terms of the representation of these other variants within primary tumor samples, you talked about the appearance of variants, and the hotspots are obviously the thing that drives the phenotype most, and then we bulk sequence. But is it known what kind of representation of all the potential variants is in primary tumors, and does that vary between different primary tumors?
Yeah, so there is some sequencing data, but generally, at least in the EGFR lung mutant cancers, they are mostly kinase domain mutations, and there are some. Okay, I should take a step back. So For some EGFR mutations, there can be compound mutations. So these mutations are not all created the same. So essentially, if you have a lung cancer and you're EGFR mutant, you're likely either an exon 19 deletion or an L858R. And then there's a whole subset of atypical mutations as well as insertions and amplifications. And so interesting, the atypical mutations can occur by themselves or they can occur as compound mutations. And so it is unclear sort of outside of those known mutations what the rest of the variants do in EGFR, but they're not all seen mutated together.
Yeah, I guess, the scene is dependent on the test, right? Other subclones, if you went back and took some of these top hits and looked, was the expectation that they'd be widely distributed? You'd have many of these examples in a primary sample?
So if we went back to look at primary samples, I assume that the well-characterized ones would show up, but the rare mutations. So the nice thing about UCLA is we see a lot of EGFR mutant patients, and that's generally due to the demographics of the region. And so we actually start to get some of these rare mutations, and we also get some mutations that aren't in bioportal that we haven't actually seen before.
I've got a question online, but I don't have my glasses on. Hang on.
I can, hold on. I saw it.
Here we go.
Oh, here it is.
Does this system work on other gene, protein, or only for EGFR?
No. So this can work for whatever gene you want to assess. So you just need something that has, ideally something that's growth or can regulate growth promotion. And so you can, and many genes can contribute to growth promotion. So you could essentially do this for any protein. So this has been done. So EGFR today is like the biggest protein that this has been done for, but there's also, people have done P53 mutations, both the ORF overexpression and the CRISPR endogenous like knock-in mutations too. And there's also, I think I read a paper, there's like a RAD51 deep mutational scanning screen. There's an IDH1, there's like a bunch of other types of proteins that you can, in fact, use this same sort of schematic with.
You have another question online. Is there a way of further screening to evaluate the interactions of mutant cellular domain downstream signaling intermediates?
Sorry, could you say that again? Oh, no, that's fine too. No, I didn't hear it was the question. Okay. All right. Is there a way to further screen to evaluate the interactions of mutant intracellular domain mutations with downstream signaling intermediates? So we could think about doing an arrayed screen, and it'd be a little bit more challenging because you have 24,000 variants, and so that'd be difficult to plate in probably your 384 well plates. But you can imagine doing like an in-cell Western, which you can do, and then actually connect growth to signaling properties. So it is very doable. It's just, I don't know of a good way outside of an arrayed screen to do that at this point. Or actually, that's true. We could also maybe tag different signaling cascades with different colors too. And then you might be able to do it through flow. Might be another way to do this, do a screen like that.
That was great. Thank you. Are there any more questions in the room or online before we thank Dr. Hayes again? Anyone? Last call. Okay. Thank you so much, Tiki, for a beautiful talk.
Yeah, thank you.